This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 220, recorded on June 25th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, here I am. Thank you for having me.
You don't have to thank me. You're a member of the team. Good to see you. Cool. Good to hear you. Good to hear you. It was about a year ago that we actually saw you. How about that?
That's right. You were here. That was fantastic.
Right after the microbe meeting.
That's right.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, hello.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And all four of us were there, right?
We were. And the planes were on time. No luggage was lost. And we had Ray. We had Ray with us as well.
That's right.
Boy, that shows you how the world can work. What a difference a year makes.
Oh, my goodness.
It's just, and it's not getting better anytime soon, is it?
At least not in Charleston. At least not in Charleston.
Nowhere.
Well, Michigan has done a great job. I credit Big Gretch, our governor.
Yeah, your cases are way down?
Yep.
And you're not spiking now that everybody's going back?
No, I mean, we've seen a small uptick, but we're way down from our peak.
Wow.
Are people going back to work slowly?
I am mostly in the house, so I'm not monitoring very carefully, but our research labs have been slowly ramped up, but, at 30% capacity. 30% capacity. So.
What about the outside world? Are things opening?
They are. Although the governor just announced that our gyms and exercise facilities will not open as scheduled.
Oh, okay.
So,
well, it's good to be cautious. It's going to take some time. All right. Today we have no COVID-19 news for you. Sorry. So I'm telling you now, if you want to leave, you will miss two cool stories. But there are other things going on out there in science, and it's time to diversify. I am going to start with a snippet, which is out of this world.
Literally. I was going to complain that you have to pick papers where I don't have to look up the words in the title to know what they mean.
Oh, I had to look it up too. Here's a paper from Scientific Reports, and the title is Methane-producing Archaea Can Produce Methane in Deliquescence-Driven Mars Analog Environments.
No wonder we're out of this world.
So I get all of those words except deliquescence. I had to look it up. It's the process by which a substance absorbs moisture from the atmosphere and eventually dissolves in it. It makes a solution. Makes sense, right?
Oh, yeah. It happens here all the time naturally in Charleston. You just have to leave your sugar out and it turns into a brick. So
That's right. That's right.
We see that happening all the time, but
it happens with salt too, right? The salt
yes.
can absorb water and melt. So yes, it's a fancy word for absorbing water and getting dissolved in it. Anyway, this is from a number of institutions, the Technical University in Berlin, the Robert Koch Institute, Tufts University. Hey, Tufts, Elio.
Hey, yeah.
Right.
How about my old haunt?
And mine.
Imperial College London, the German Research Center for Geosciences, the University of Potsdam, and the Leibniz Institute of Freshwater Ecology and Inland Fisheries. The first author is Maus, Heinz Schermack, Iro Nuaves, Wagner, and Schultz Makuch is the last author. So this is something different. It really actually was sent in by a listener.
I was wondering how you got to this. This is not usual, is it?
It's not my usual beat, right?
That's right.
No.
In fact,
it didn't have SARS in it. It didn't have SARS-CoV-2.
I'm taking my hat off to you for tackling something like this. Pretty gutsy.
Oh, I think it's okay. have you guys to help me, right?
That's right.
Oh boy, we're the experts.
This was sent in by Amir, who said this was a really cool paper. And Amir is in Jena, Germany. Okay, so and this study is largely out of Germany. Okay, so here's the story. Very cool. So Mars has methane in the atmosphere that was first detected by a probe rotating the orbiter, the Mars Express orbiter, and then later the Curiosity. Remember the rover? It went down on the surface.
Oh, yeah.
And they detected it too, but it's parts per billion. And so that's not a lot. And it fluctuates throughout the seasons. So they're wondering, the idea is emerging that reservoirs beneath the surface of the planet are sporadically releasing methane, because there's no way they think it can be maintained in the atmosphere. It's just too much UV and so forth. And so they say maybe it's released, or maybe microbial processes beneath the surface of the earth are making methane, right? Now, the problem is there's no water
at the surface. But remember, we now know from South Africa that bacteria can grow many miles below the surface of the earth. So there may be water many miles below the surface of Mars, and gases always find their way to the top.
They do. So their thinking here is that maybe there's subsurface salts, hygroscopic salts that absorb water. This is the deliquescence, right? And that provides the water needed for microbial growth. And that is what they set out to test in this paper. They made what they call a closed deliquescence system. It's basically a big bottle, and they have a container in the bottom, which it contains soil, and they have a approximated Martian soil, which I'll tell you about in a moment. And they have microbial cells, archaea, and with or without salt, right? The salt will absorb the water. And then around this container, they have saturated potassium sulfate, which is going to humidify the atmosphere so that the salt in the dirt can pick up atmosphere. Okay, so this is their artificial deliquescence.
I too was so surprised by this really simple apparatus after having to use a dictionary to get through much of the first page of the paper.
Oh, yeah. I mean, it looks like my sugar bowl on my kitchen table. So those of you who haven't seen the paper, it really looks like your sugar bowl on your kitchen table.
It's elegant. It's elegant. Let's not take away from that.
No. This is a brilliant design.
So a little more background. The experiments are rather straightforward. So there are similar environments on Earth, and they have microbial environments. For example, there is deliquescence and microbial communities in the Atacama Desert of Chile, the McMurdo Dry Valleys in Antarctica, and the Larsman Hills in East Antarctica. And these have been studied to try and get some information about Martian environments. And there are extremophilic microorganisms that have been adapted to such environment on Earth, particularly methanogenic bacteria. They use carbon dioxide and hydrogen as an energy and carbon source, and they make methane. It's called methanogenesis. And if I remember correctly, these were once thought to be bacteria, right? And then they were found by Carl Woese to not be bacteria. They were archaea. And then we have the soil. And they have tried growing these methanogens on what they call Martian soil simulants. And here's another name that I had to look up. Martian regolith analogs.
Yes. We all looked up regolith.
We did.
So this is Martian dirt, basically.
Martian dirt. That's what it boils down to.
You know, they have sent rovers to Mars, and they've gotten samples of dirt. So we sort of know, what the dirt's made of, and its texture and so forth. So
is it red?
Oh, yeah. It's got a lot of iron in it. Yeah.
And so people have that's what a Martian regolith analog is. It's an approximation of Martian dirt. Of course, it's made here on Earth. It's not coming from Mars, but we put it together to approximate. And then they've tried to grow methanogens in it. But as they say, no one has tried to find out if the methanogens will grow beneath the surface, and the only water present is made by absorbing it from the atmosphere, right? So that's the deliquescence part. So that's what they set out to do. So they took 3 methanosarcina species, and they have different compositions of the dirt. And the key control here is to leave out the salt, because without the salt, the water won't be drawn in, right? And that's what they want to know. Can the salt draw in the water enough so that the archaea will grow?
And remember, a sarcina shape is effectively round. So that gives you an incredible surface area to mass ratio to absorb any of the needed water in order to conduct the chemistry. So that's probably why they picked a sarcina-like methanogen.
I love the name of one of them, Soligellidae. You know what that means? It means frozen soil. Species name.
Soligellidae, yeah, that's right.
We have all been using our dictionaries.
So anyway, the first experiment, they put these 3 in the Martian soil approximation, and if they used either quartz sand alone or a Martian regolith analog from the Johnson Space Center, they didn't get any methane made. So they're measuring methane production in this jar. But when they use the Martian regolith containing phyllosilicates or phyllosilicates, then they get methane.
And phyllosilicates, as we all looked up, means sheet silicates.
Sheet silicates.
In the shape of sheets.
And they get substantial amounts of methane produced when they do that. So again, you have salt in the soil that's drawing off the water, and the control is no salt, you have no methane produced. That's the key. And they actually monitor the absorption of water by simply weighing the dirt to see how much water has gone in. They show that with salt, you get water absorbed into the soil, so that's good.
But everyone who's taken freshman chemistry has done that experiment of weighing to see whether your water has disappeared. That was the famous crucible experiment where you would keep putting that damn thing into the oven, drying your chemicals off.
So here's some of the details. M. Soligellidae. Is that Italian, Elio? Latin, okay. M. soligellidae began to produce methane after 28 days at 4 degrees in this Martian soil with 30% sodium chloride. And they got to 0.2% methane after 64 days. The negative control with no salt showed no methane production. Another archaea, M. barkeri, no methane at 4 degrees, but did make it at 28 degrees. And M. mazei didn't make methane any time at all. So the strains are different. It's quite interesting. So that's really it. That's the experiment. You can have rehydration of soil, and it will. By the way, these bacteria were desiccated when they put them in the soil.
Right, brought them back to life.
And so they are brought back to life. Yes, by the water being absorbed.
The Martian-like.
Martian-like. That's right.
And it should be pointed out that their control is they add perchlorate to the medium, and perchlorate will inactivate things, and
does that kill the archaea, basically?
Principally, yes.
Yeah.
So it's their control.
That's a good control. So the making of methane depends on the species of methanogen, the incubation temperature, and the type of soil and the salt that you use. And the phyllosilicate, of course, is important, as they show. They say it has a high capacity to retain small amounts of water, and it's also known to be used as nutrient sources for methanogens. And a little more information on these strains. M. mazei was isolated from a sewage sludge in California, and that's one that didn't have any methanogenic activity in this system, but it is the closest relative to M. soligellidae,
which was isolated from permafrost in Siberia.
Permafrost in Siberia. It was only active at 4 degrees, and maybe that's why it's adapted to that. And M. barkeri is from a sewage sludge in Illinois, and that only worked at 28 degrees.
Which makes sense.
All right. So they can make methane in what looks like Martian soil by deliquescence. So they say it's a model organism for studying how microbes could thrive in Martian environments.
It's exciting.
And Michael mentioned also deep subsurface.
Yeah.
They can also live there as well.
Well, I mean, if you were going to think about where the microbes would be on a planet like Mars, we all know that you absolutely need liquid water, and the likely location for the liquid water is well below the surface. And that's where I would look is, drill a well and see if you can find them down there. And the gas is going to percolate up through pores in the soil to go up to the Martian atmosphere. And, depending upon the warming and movement of I don't know if Mars has tectonic plates, but that would effectively, liberate water or move the water around. We could effectively see what's going on. And in fact, they suggest, the putative subglacial lake near the Martian South Pole could be supplied with hydrogen through serpentinization reactions, in turn feeding the methanotropes in order to generate the methane, because you need the hydrogen as the electron donor in order to make the methanogens, in order to make methane, I should say.
Yeah.
I was really impressed with how hardy these archaea are. They can tolerate UV radiation, freeze thawing, really high pressures, and also up to 3 weeks of the thermal conditions that are expected on Mars, are known to happen on Mars. So this is a tough nut.
Mm-hmm.
So the question then becomes, what would you expect on Mars if life is there? Would you expect it to be an archaea, or would you expect it to be a true bacteria?
Right now, I'd say the archaea are leaders in the clubhouse based on this paper.
I think so, given these results, right?
Yeah.
Yeah.
But how did they get there? Maybe they were always there. Maybe they evolved independently of Earth, right? I mean, that would have to be.
Yeah. They may not even be archaea. It was sort of the red herring question.
Maybe it's something totally different.
Yeah.
Who knows?
Maybe the even more challenging scenario would be to imagine that that Mars was sterile. That's even harder to imagine, right?
Well,
that something wouldn't have
No,
evolved.
I put my dime on that.
Do you?
Yeah, I'm not a big one about life in outer space.
Evolution couldn't solve it with enough time?
I don't know. I think life is so unlikely.
Well, and to give the homage,
we can talk about that forever. That's a big argument.
To give the homage to Mark Martin, it could have been a tardigrade that brought the archaea to Mars.
I was thinking today, at some point there's going to be a human colony on Mars, probably, right? And they're going to have to live in structures and wear spacesuits outside because the atmosphere is not good enough, right, to breathe. And we won't have – the only viruses we'll have are the ones we bring with us.
Yes.
Because there are no bats, there are no other animals to give us viruses. So it'll just be – as long as you send people there who are healthy, you could have a pathogenic virus-free planet. That could be interesting, right?
I bet I put my money on that.
I guess not. Nobody sounds excited about that.
I wouldn't be signing up for that a stint up there. No way.
No, I wouldn't either. I would not, because
they have
getting that back is dangerous, right?
Well, they have no protective shield that we have on Earth. They don't have the molten core that generates our magnetosphere that prevents us from, feeling the effects of cosmic radiation to the level and extent that it's seen on Mars.
So we had Hazel Barton on TWiM a long time ago.
Oh, yeah.
And Hazel likes to explore caves for microbes. And I said, if you could, would you go to Mars? And she said, right away.
Wow.
There's a brave person.
She's great.
She is unique.
Okay. Did you like that snippet? I thought that was fun.
I did. I thought it was a really great choice for a snippet, too.
Thank you, Amir. Great pleasure. Amir sent it in April, which seems like forever ago.
Eons ago.
We appreciate it. Okay, so now on to our paper, Michele.
And we're moving from Mars to the vagina. The title, just to show you our breadth, the title is Identification of a Cytopathogenic Toxin from Sneathia omnii, and it is from a group at Virginia Commonwealth University. Gabriella Gentile, Amy Rupert, Lizette Carrasco, Aaron Garcia, Naren Kumar, Scott Walsh, and Kimberly Jefferson.
Quite a paper it is.
Yeah. So this group is studying a bug that you might not have heard of. And in fact, it was only discovered in the 2002. There was a 27-year-old woman in the second trimester of pregnancy. She had fever, headache, pain, came in, got worked up, didn't find anything. But then on her next appointment, the fetus had lost its heartbeat and was fetal demise. So they collected some of the amniotic fluid and found that it was turbid, discolored, foul smelling. And they were able to isolate an anaerobic coccibacillus that upon subsequent sequencing and phylogenetic analysis was identified as a new species of a new genus. So this is Sneathia amnii. Sneathia amnii.
By the way, Sneathia, it's after David Sneath, who's the guy who made up quantitative taxonomy. He's the author of what we now do in taxonomy. Sorry about it.
And then amnii refers to, of course, the amniotic fluid where they first discovered this bug. So Kimberly Jefferson's lab has been studying this for some time. They published the whole genome sequence in 2002. And then at Virginia Commonwealth, they have formed a vaginal microbiome consortium that is funded through the NIH Human Genome Project and have been able to do a lot of really sophisticated genomic analysis. And the focus has been mostly on genomics because this is a difficult to culture organism. We don't yet have genetic mechanisms to manipulate the bug. It's got a very small genome and it's difficult to culture. And yet, in a longitudinal study that they did at Virginia Commonwealth University, they found that up to 40% of women who came in, were part of their study, had this particular bug in their as part of their vaginal microbiome. And it's been associated in a number of studies with complications of pregnancy, sexually transmitted infections, cervical cancer, and also infections of the male urethra. So clearly we need to know something about it. Worldwide, 10% of pregnancies don't go to full term. So 15 million preterm births annually, that it's a tragedy. Often this is poor outcomes for the fetus or the developing child. And it also accounts for $26 billion in healthcare costs annually. Another interesting feature that this group has discovered is that women of African descent are at higher risk of preterm birth, and they also are more likely colonized or have a different microbiome than women of European descent. So this group obviously was motivated to understand more about this pathogen. So far, no virulence factors had been described, but they had noticed in an earlier study that when they co-cultured this microbe with cells from the fetal tissue or from, like the amnion or the chorion, which is the sac that is around and protects the amnion, that they saw some cytotoxicity, some cell rounding. So what they do here is really a classic approach to identify a toxin. So in their first experiment, they take samples of human chorion tissue from human deliveries that have gone full term, and they co-cultured either with E. coli, garden variety E. coli, or with this Smithia. And what they saw is that the membranes, both on the apical and the basal surface, clearly got damaged. They were permeable to trypan blue, suggesting that this bug was able to damage the surface but also traverse this tissue. And similarly, they did cytotoxicity assays with two different cell lines, human amniotic epithelial cells and chorionic trophoblasts, and they also saw similar cytotoxicity. And they could demonstrate that this microbe would lyse red blood cells. But curiously, only if the red blood cells were from humans or cows, but not from horse or sheep or rabbits. And that's shown in figure 2. So that's a clue that perhaps this bug is binding to a specific protein that's unique to humans and cows. So it might be a receptor-mediated process. So they wanted to understand what was causing the cytotoxicity. And again, they don't have genetics available just yet. So they took a wise bioinformatics approach. They had the whole genome sequence. So they just did a search. Do we see anything that looks or smells like a toxin? And while they didn't see any classic toxins, they did find some intriguing similarity to what's called a two-partner secretion system that is known to equip other gram-negative bacteria to deliver toxins into host cells. And in fact, this one had homology to a well-described toxin of Bordetella pertussis called filamentous hemagglutinin adhesin, which is a component of the acellular vaccine that we use. So this clearly was a really tempting candidate, and they decided to pursue it. So what they did is first just clone a fragment of this. Let me tell you too, these two-partner secretion systems are very convenient because one partner does the job of getting the target protein across the membrane. So they are genetically linked on the chromosome, so you could easily find the toxin and its secretion partner. So they cloned part of the toxin and expressed it in E. coli and made antibodies. And that was useful because then they could ask if these antibodies bind and block activity of this particular protein, does that now inhibit the cytotoxicity? And in fact, that's what they saw. So if they pre-incubated the bacteria with these antibodies and then added them to their target cells, they no longer saw the cytotoxicity, whereas the control serum had no effect. So again, a really good indication that they were on the right track. So then they went to great lengths to purify this beast of a toxin. So it looked like, based on some earlier experiments, more than 200 kilodaltons membrane associated. In the paper, they make it look like it was quite simple to get their hands on this. But Elio, I think you know the backstory.
Yes, I will. I'll tell you a little bit later.
Okay. So they then, again, developed a tool to facilitate their studies. They took the full-length protein and they added an epitope tag, a string of histidines, which are useful as a way to grab on and purify the protein from a lysate by affinity chromatography. And then they went back to their cytotoxicity assays and asked whether this full-length toxin prep could lyse human red blood cells, and it did. Likewise, it damaged these chorionic trophoblasts as they had seen for the whole bacterial cell. So again, they're narrowing in. They found the protein, have demonstrated it's cytotoxic as a pure toxin. So they then did a very elegant series of experiments to ask if it was a pore-forming toxin. So this is a classic approach where you take advantage of the kind of physics of host cell lysis. It's basically an osmotic shock. So if you balance the osmotic pressure of the cytosolic contents with the extracellular media, by adding polymers, you can block the lysis, even in the presence of the pore-forming toxin. So in particular, they use polyethylene glycol, which is convenient because you can buy it in different sizes that would then have different diameters. So the polyethylene glycol that can get into the cytosol would act as a balance and prevent lysis, whereas the larger ones would not, and you'd get lysis. So that the results of that experiment,
I think that's really quite elegant, isn't it?
Isn't it lovely? It's classic, and the figure is beautiful. I would have been so proud if this were my data that I plotted. They show that when they add polyethylene glycol between 1,000 and 2,000 species, they could partially or fully block the lysis. And therefore, because they knew what they call the hydrodynamic diameter of these different polyethylene glycol molecules, they could deduce that the pore size formed by this newly described toxin is between 2 and 2.8 nanometers. So that, in a nutshell, is the paper. And it's exciting because it is the first description of a toxin and a virulence factor for this microbe. And it now can give them a handle on screening vaginal secretions, not only for this microbe, but for the toxin itself. And this could become a tool to better screen women during pregnancy and to identify if they are carrying this microbe and if it's toxigenic, and then identify these people that might be at higher risk of preterm delivery and therefore manage their case to protect them. And in fact, they have shown that these microbes are sensitive to a number of antibiotics. So I think...
do you think that the antibody could be useful to prevent damage in a pregnant woman?
It could be short-term, but probably... Yeah, a vaccine. Yeah, the antibody, of course, they'd have to have multiple deliveries, treatments over the course of the pregnancy. But I think it does show that the Human Microbiome Project is bearing fruit. I guess bearing is a little bit punny in this context. Michele. But and it's wonderful that there's this focus on the vaginal microbiome because of the really large health burden of these preterm pregnancies worldwide, including here in the U.S. So it's an example of going from genomics and looking at the population of microbes in the vagina down to classic molecular biology and toxin assays to begin to understand the pathogenesis of one bug that is correlated with a lot of poor outcomes and even cervical cancer.
Michele, you said that 40% of women are carriers. Is that right?
Yeah, they did a screen in this longitudinal study of more than 700 women who had come in for either routine obstetric or routine GYN care or pregnancy care. And 40% of the vaginal samples had this microbe, Schnittii amnii.
Are they thinking that the ones who have problems are those making the toxin, or?
So that's possible. You know, of course, the toxin hadn't been known, so that we haven't been able to do those studies. They do describe it as an opportunistic pathogen, and whether the bug alone is sufficient, or you have to have certain numbers, or the population is out of balance, and you have colonization resistance or not. I think those are all really good questions. And then they also, to their credit, identify a number of kind of protein chemistry questions. You'll see from the protein gels that this purified protein migrates as a whole suite of species, some as small as 40 kD and then going up to 200 kD. So they're imagining that it could be processed, or there could be modifications to the protein. There's still a lot to understand about whether it functions as a monomer, a ligamer, etc. But they clearly have got their hands on the first toxin described for this opportunistic pathogen of the vaginal microbiome.
You mentioned that men have it. Do we know what fraction of men are carrying it?
I don't think we do. I haven't looked deeply in that, but it's definitely a number of papers have associated with sexually transmitted infections and with male urethra infections.
That's why I asked earlier about the vaccine, because you would likely want to test the vaccine in men so you wouldn't run the risk of a fetus dying should it have unforeseen consequences in the vaccinated population. But I would think that if you do a simple make a toxoid of the toxin and try to immunize men and ask the question, do they still carry it? And does it indeed then profoundly change their urethral microbiome? Because that may be one way we could safely evaluate the vaccine construct, albeit in men, before we take it to women, where we then, of course, run the risk of pregnancies. And you don't want to run that risk of the vaccine having an unforeseen consequence in the developing fetus.
Yeah, that's good thinking, Michael.
Okay, so let me tell you a little bit about the senior author, the first author, Gabriella Gentili, or Gabby, as she's known. We contacted her, asked her a lot of questions, and she told us a lot about her. So she's currently a graduate student at the University of North Carolina in Chapel Hill, working in the pharmacology department on the antibiotic resistance of gonorrhea, under the mentorship of Robert Nicholas. But before that, she was an undergraduate at the University of Richmond in Richmond, Virginia, and then where she did some research on freshwater sponges and their symbionts. And she liked it so much. In fact, she had a passion for science that goes back to high school or earlier, watching TV shows and movies with scientists turned her on. Especially she liked Abby from NCIS. I don't watch NCIS, but she was totally taken by that. So anyhow, she was exposed to research as a freshman in college and ended up applying to an NIH-funded program called the Postbaccalaureate Research Education Program, or PrEP, at Virginia Commonwealth University. And this gives recent college graduates from groups which are underrepresented in scientific research the opportunity for immersion in research for 1 year. So the PrEP program did it for her, and she started to work at Virginia Commonwealth under Kimberly Jefferson on the project of this paper. So the groundwork for this work was done by another master student in the Jefferson lab, Amy Rupert, and she was able to express the Schnitzia amni protein, CPTA, and made an antiserum. The antiserum, of course, was very crucial, as you can tell from the paper, and it inhibited the activity, the hemolytic activity of S. amnii, suggesting that it may be a hemolysin. So her job was to purify CPTA, because without purifying it, you couldn't go very far. Anyhow, that was not easy. That took a lot of work. So she tried using Western blot to determine that it's there. And she says that the most exciting day in the lab was when she finally purified CPTA. And this was from the outer membrane of S. amnii. They've been trying forever to purify it, and nothing worked. So finally she used a functional assay. She kept looking and looking until finally on a Western blot, what looked like she says, what seemed like my 1,000th cation exchange chromatography experiment. And after that, she found a band, and she left the film in Dr. Jefferson's desk for her to look at, just to make sure that this was right. And she writes Later, she came over to my desk, and I could tell by the look on her face that we had finally gotten it. So she did, and that was the first discovery she made on her own, an important discovery that led to her being the first author in this paper. So
let me just interject, Gabby, that it felt like you did 1,000 of these ion exchange columns. That's why we call science research and not search. We redo it and we redo it and redo it.
Very good. She did research. Okay. So anyhow, she sounds like a very dedicated person, an extraordinarily competent one. When she's asked to what advice and encouragement does she have for junior colleague, she says, don't be afraid to ask questions for help. Not knowing the answer or how to do something in the lab is completely normal and okay. Asking for assistance doesn't make you look dumb. It's the only way you're going to learn and grow to be a scientist. This is something I still struggle with, and I have to remind myself every once in a while that questions are good. The scientific community can be intimidating, but it's important to be confident and ask for help so you can absorb as much information as you can. Amen, Gabby.
So wise. Isn't that exciting that her Postback project led to a first author paper? And I think I failed to say the Journal of Bacteriology. And this was just published in June this year.
Yes.
That just saves you from getting a letter from Tom Silhavy.
Nice. Oh, I always ask for help. I have no problem with it. I guess it's something you have to learn, right?
It's the most
But
important lesson.
I know what she means. Look, I know what she means. You feel vulnerable. You feel like, gosh, you ask too many questions. Maybe you don't belong there.
Or I'm supposed to know this or that.
It's a valid point.
It'll pass. Yep. It'll pass. Don't worry. Wonderful. Thank you, Michele, and thank you, Elio. That was great. That's it for TWiM 220. You can find it at microbe.tv/TWiM. That's where all the show notes are and all the letters. If you want to send us a letter, TWiM at microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Today on TWiV, everybody's here. Michele Swanson from the University of Michigan. Thanks, Michele.
Thank you, and everybody stay safe.
Elio Schaechter is at Small Things Considered. Thanks, Elio.
My pleasure, as always.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. And remember to wear your masks and wash your hands.
Oh, yes. Wear your mask, wash your hands. And if you can stay home, do that too. I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.